Such a large part of what I do these days is interrupting good conversations, so I'll do that yet again. So good afternoon and welcome to our Fall Professor Laureate Lecture and Annual Teaching and Mentoring Awards Ceremony. This is a time for us to gather and honor a few of our colleagues for their impressive achievements and contributions to their discipline, the college, and the university. We will first recognize the recipients of the Teaching and Mentoring Awards and then the Dean's Recognition Award, and that'll be followed by our Fall Professor Laureate Lecture from Dr. Eric Ross. And of course, I hope you will stay for the reception that will follow the ceremony. I'm now pleased to introduce Executive Associate Dean Simon Taverner, who will present the Teaching and Mentoring Awards.
All right, thanks, Jake. As happens every year, the accomplishments of this outstanding group are so numerous and varied that it's impossible to acknowledge them all in finite time. I'm therefore forced to fall back on presenting the items that resonate most with me as being especially interesting and noteworthy. I hope that you'll be able to imagine the rest and will be convinced that their other activities meet the highest of standards. So, I'd first like to present the Faculty Excellence in Undergraduate Teaching and a Mentoring Award to Aaron Nielsen. As you're probably aware, I'm enthusiastic about course and program development and providing students with a modern and relevant education. Dr. Nielsen has developed the undergraduate certificate in sports statistics and analytics. I know from multiple informal interactions with students that these courses are tremendously popular. The certificate comprises two new courses, Stat 351 and 451, which are Sports, Statistics and Analytics 1 and 2, and one course from a menu of 300 and 400 level courses, for example, Bayesian data analysis. Aaron is also an active mentor of summer undergraduate research projects with such titles as Informing Tactical Periodization in Collegiate Football, Analyzing Spin Direction for Division I Softball, and Playing the Odds, Defensive Positioning Strategies to Minimize Batting Averages in Major League Football. Now, baseball. Well, you know, what do I know about football? Sorry about that. Now, if I make a small digression, I know you're all dying, as I was, to know what tactical periodization is. So naturally, I went and asked Copilot. So, it is a training methodology that integrates technical, physical, and tactical elements, treating them as an inseparable part of a larger game model. Okay, so that's your classic word salad, right? The next is, instead of isolated drills, training sessions are designed to mimic the specific tactical principles and intensity of a game, with a focus on developing players' ability to make collective and individual decisions under pressure. Okay, so I sort of understand that. I'm also really interested in effectivity. So two of Aaron's advisees now hold coveted positions as data scientists with the Cincinnati Reds, which I understand is Major League Baseball, and Houston Dynamo, which is Major League Soccer. So, Dr. Nielsen, please come on stage to accept your award. Congratulations. Oh. All right. Congratulations.
Thank you.
Our next Faculty Excellence in Undergraduate Teaching and Mentoring Award goes to Dr. Harmony Tucker. Dr. Tucker has been the driving force behind the transformation of the chemistry department's approach to teaching general chemistry. When she joined CSU in 2004, the sections of general chemistry were separate entities unto themselves and operated with little or no coordination. Students had very uneven experiences across sections. Why am I not surprised? I'm sure that this also manifested itself in their performance in later courses. Dr. Tucker has dedicated her career at CSU to improving that experience. Let me perform a rough estimate of enrolments in Chem 111 and 113 over the last 20 years. Let's say Chem 111 enrolls 2,000 students per year and Chem 113, 1,000. So I'm using powers of 10 here for the physicists in the room. Over 10 years, again for the physicists, that's 30,000 students, and over 20 years, 60,000 students. For those who quibble, physicists again, that many students take both courses, and so these are not 60,000 individual students, let me consider student credit hours. At 11,000 student credit hours per year, one's a 4-credit course, one's a 3-credit course, or 220,000 student credit hours over 20 years, and a notional $500 per credit, that's 110 million. She's underpaid. Now, I realize this is a really fanciful calculation, and this must be attributed to the efforts of many, many other people, but it does indicate the importance of these courses for both the students and the financial health of the university. Oh, actually, since you're interested, at this point, I'm going to refrain from how many years this represents of the football coach's salary. But seriously, though, chemistry represents a significant challenge for a large fraction of students who are now on campus or who have graduated from CSU. Judging by their responses, students have clearly appreciated these changes and Harmony's own structural expertise. Through improving student success and their appreciation for chemistry, the college and the university owes Dr. Tucker a huge debt of gratitude. Dr. Tucker, please join me on stage. Congratulations. Oh, no, thank you. Thank you. Our next recognition is the Faculty Excellence in Graduate Teaching and a Mentoring Award for Dr. Shradeep Palakara. I need to begin by saying that I'm a big fan of Dr. Palakara and his accomplishments, not only in terms of graduate education, but in all aspects of a faculty member's professional life. It's therefore a great honor to be able to recognize him in this way. Rather than simply quoting the numbers, I'd like to look at outcomes. Together with a large array of master's, PhD, and REU students as co-authors, Dr. Palakara has published over 100 peer-reviewed papers in top-ranked venues. His students have received Best Paper Awards and the IEEE Technical Community on Scalable Computing Best Dissertation Award. One of his former students, now a faculty member at the University of Washington, has received the prestigious NSF Career Award. This is a testament to the time and care Dr. Palakara invests in his graduate students, meeting them individually twice a week and holding project-specific group meetings once a week. He has created a lab culture of respect and curiosity. While time and effort mentoring graduate students is essential for success, there must be more. I think it is Dr. Palakara's approach to science that he instills in his students. Science is not simply solving an isolated problem and moving on. In his own words, it means approaching problems with rigor, analyzing data with care, presenting findings with clarity, and drawing conclusions that hold up under scrutiny. I think this is something on which we can all agree. Dr. Palakara, please come to the stage to receive your award. I will now present the Early Career Faculty Excellence in Teaching and or Mentoring Award to Dr. Megan Hill. Dr. Hill helped chemistry graduate students establish the American Chemical Society, and it's Poly/PMSE, which I'm going to explain in a minute, student chapter at CSU, and she remains the faculty advisor. These student chapters represent the Division of Polymer Chemistry and the Division of Polymeric Materials, Science and Engineering. This student group was recently recognized by the American Chemical Society as the best such student chapter in the nation. These students organized a full-day symposium entitled Elevating Polymer Science in the State of Colorado and included a career panel. Megan serves as the co-director of the CSU Chemistry REU program, and as one measure of its effectivity, several students have come back to attend the PhD program at CSU. The importance of science communication has become perhaps an existential one in the current social and political climate. Dr. Hill, together with Kate Wilsterman in biology and Colin Rice in philosophy, have explored multidisciplinary science communication and organized a 2-day workshop, including a podcast hosted at CSU. The NSF-funded center that Dr. Hill runs, the Center for Sustainable Photoredox Catalysis, has hosted multiple workshops, creating presentation templates for community Science on Tap events, and supported students in presenting to non-scientist audiences at these events. Dr. Hill, please come and join me on stage so I can present you with your award. Congratulations, fantastic work. Thank you. Next, I'm excited to present the Graduate Student Excellence in Undergraduate Teaching and our Mentoring Award to Alexa Jane. It's my personal opinion that the psychology department asks rather too much of their graduate students, but I'm continually proven wrong, and I'm astonished by the amount of high-quality work that graduate students in psychology perform, and with such evident passion. Amongst this very special group, Alexa stands out. She has been the instructor of record for 7 distinct classes, from Psych 100 to undergraduate research course, and has truly taught across the curriculum. She has developed 300 and even 600 level courses. The graduate level courses are Psychology 621 and 623, which are the psychology of calling and meaningful work and positive education. From the catalogue for Psychology 623, this course is for people interested in educational systems that teach academic content and also help students develop the best potential and cultivate skills for well-being. Sounds like something we might all take. So, Alexa has received literally hundreds and probably thousands of outstanding teaching evaluations. Why do I say that? Well, as one example, she received 102 positive responses from one section of Psychology 325 alone. This is from a class of around about 150, representing an astonishing 2/3 response rate. Alexa is unable to be here with us tonight, so Dr. Marty Amberg will accept the award on her behalf.
Oh, I guess. Thank you.
Our final teaching and mentoring award this evening is the Graduate Student Excellence in Undergraduate Teaching and or Mentoring, presented to Beth Whitman. What makes Beth's nomination stand out to me is her success within a wide range of non-traditional roles. One that I am sure has had a positive impact on both students and faculty is her role as accommodations specialist, assisting faculty with student accommodations and ensuring compliance with university policies. We're all aware of the importance and increasing in frequency of these accommodations. In a related activity, she has assisted both students and faculty as a course accessibility teaching assistant, assuring, for example, that all course materials include alternative text. Again, We all know how important this is, and yet how intimidating the process can feel. In partnership with the Amplify Learning community, she's co-designed and taught Natural Sciences 170, Perspectives and Communication in Science, a first-year seminar course focusing on how personal experiences, identities, and perspectives influence the scientific process. Beth is the lead designer and co-instructor of BZ 629A, Recognizing and Addressing Oppression in the Sciences, a course for graduate students reflecting on the history of science and its impact on the present scientific culture. This course has been attended by students from across the college. Her expertise has been recognized through multiple invited guest lectures, both inside and outside CSU, including CU Boulder, Front Range Community College, and Larimer County Natural Resources Department. Beth, can you please come on stage so I can present you with your award? Right. Let's applaud all our winners again. Now I'll hand over the mic to Dean Roberts, who will present the Dean's Recognition Awards.
So I want to thank everyone who was part of the nomination and selection process for the Teaching and Mentoring Awards. We would not be able to celebrate these extraordinary members of our college without your dedication and service. I'm now excited to present the Dean's Recognition Award, which celebrates the exceptional contributions of our administrative, professional, and state classified employees. This award honors individuals whose efforts have had a significant impact not only within their own units, but also across the broader college and university community. The selection process is based on both formal and informal feedback received by the deans throughout the year. We received multiple nominations this year, each highlighting deserving candidates. Tonight, we are proud to recognize two outstanding individuals whose dedication, hard work, and willingness to go above and beyond have greatly benefited the college. Their commitment to excellence has truly made a difference, and we are honored to celebrate their efforts. We are pleased to award the 2025 Dean's Recognition Award to Simon Tavener, Executive Associate Dean, and Lisa Streep-Case, Director of Communications. Simon Tavener has served as the Executive Associate Dean in the College of Natural Sciences for 13 years, while also being a professor in the Department of Mathematics and a former chair of that department. As part of his regular duties, Simon has contributed to the success of the college in many ways. Today's Teaching and Mentoring Award event is but one example of Simon's efforts, as he has a primary responsibility for overseeing the awards process for almost all of the awards given today. He manages teaching resources of the college through distribution and management of graduate teaching assistant tuition support, oversees budgetary distributions in the summer that support having many offerings for our students, is a college representative and advocate at university-level graduate student affairs in many different respects, meets with and coordinates among the many graduate program leaders in the college in the departments. He has undergraduate curricular and programmatic leadership in many, many different ways. And this list could go on and on for a long time about his regular duties, but actually separate from these formal duties, Simon provides very useful insight and advice. I truly appreciate the fact that he does so. And while all these things are important, they're actually part of the motivation for this award recognition today. Simon was instrumental in the creation and growth of our data science undergraduate major in the college. From its curriculum to the faculty that we have who are associated with that program, to its management, to advocating for the program, Simon has been either a major contributor or the contributor to these efforts. The program has now grown to about 150 students over the past 6 years or so, and there are faculty connections to data science at departments across the college, including in ones that have not traditionally been associated with data science. I think that these efforts are worthy of recognition, and I'm happy to do so with this award. One other element of Simon's contributions I wanted to recognize was his willingness to step up into needed roles when asked. These have ranged from very large roles, such as interim dean, to other leadership roles, to willingness to serve as a temporary representative of the college at a meeting or event. I truly appreciate Simon's contributions and his dedication, and I am pleased to provide some measure of recognition for these efforts. Congratulations to Simon for being named as one of the Dean's Award recipients for 2025. Congratulations.
Thanks, Jay.
You got it, Leah. Lisa Streep Case serves as the Director of Communications in the college and has been in that role for about 6 years. I've had the chance to work with Lisa as a former chair of the Department of Physics, and my high opinion of her abilities has only grown over the past year as I've had a chance to work with Lisa in a new role. I consider myself fortunate to be able to rely on Lisa's work and talents whenever there is a need for communication in a standard or especially unexpected situation. The quality of Lisa's work is excellent, but similar to Simon, there are some special contributions in this past year that were a large part of the motivation for this recognition. There have been lots of firsts, or at least it's been a long time, events in this past year. First, and probably foremost, is since late January, the landscape of higher education in the U.S. has been changing rapidly and in numerous ways. While all of us in this room have been affected, Lisa has had to adapt to changing circumstances in a rapid fashion numerous times in response to these changes. Another first, the Board of Governors now has an annual summary that is to be provided by every college. Due to a quirk in scheduling, we had relatively little time to put the CNS presentation together, and Lisa's work with respect to this presentation was exemplary. I really wanted to develop themes that we could use to communicate about the wide range of scientific breadth across the college, and Lisa has suggested and developed a framework based on the broad themes of life, data, and matter that has received many positive comments when presented in wide university settings. We're just concluding the search for a new permanent dean. I think the deadline's like now or something, maybe a little bit. And Lisa played a leading role in the preparation of the prospectus and communications about the college, which is no small amount of work. In addition, Lisa has pioneered and implemented a new college-level recruiting effort that focuses on improving the yield of students who have applied to CSU with an interest in one of our CNS programs. These are students who are choosing between different institutions, and we have the opportunity to help them see all of the really good things that CSU has to offer. I can speak from experience, both as a former chair in a department and maybe a little bit as a parent of college-age kids, but please don't tell them that I mentioned this, that there are not easy ways to break through all the messages that they get, and Lisa's done a great job of creating messaging by current students that connects with potential future CSU students. I'm very excited by these efforts, and we continue to develop new ideas and goals as we learn more. Recruiting will be increasingly important as a priority for the college and university, and the new recruiting effort pioneered by Lisa is not only an excellent activity in general, but one that is very timely. Lisa, thank you for all that you do in the college and the university, and congratulations on being named as one of the Dean's Award recipients of 2025. Lisa, if you could please join me on stage. So please join me in a final round of applause for all our award recipients. The Professor Laureate title is reserved for faculty who inspire their students, make extensive contributions to their field, and devote time and energy in service and outreach to the university, the profession, and the community at large. It is the highest honor the college bestows on our faculty. Recipients of the award hold the title for 3 years. Each is awarded an honorarium and a small stipend during each year of their term for student research support. This past spring, the college named 2 faculty members as Professor Laureates, Dr. Delphine Farmer from the Department of Chemistry and Dr. Eric Ross from the Department of Biochemistry and Molecular Biology. Dr. Farmer presented her Professor Laureate lecture in the spring, and this afternoon we will hear from Dr. Ross. Before the lecture begins, let me tell you a little bit more about Dr. Eric Ross. Eric has been at CSU for 20 years, and in that time, he has made significant and consistent contributions in research, teaching, mentoring, and outreach. His colleagues describe him as a thoughtful leader and a well-rounded faculty member who shows excellence across all mission areas. Eric is an infectious disease researcher who specializes in prions. Prions are infectious proteins that are distinct from viruses and bacteria. They cause proteins to misfold, causing various diseases such as chronic wasting disease and mad cow. Tonight, Eric will take us into the world of prions and infectious diseases with his lecture titled Yeast Prions as a Model for Amyloid Diseases, How Questioning Assumptions Has Revealed the Unexpected. Eric?
All right, so first off, I'd like to thank the college and the dean for this amazing honor. I'm looking around at the fantastic faculty in this college. It's incredibly exciting to even be nominated, much less to win an award like this. So I apologize for the really long title that I made you read, but it highlights the two major goals that I hope to accomplish over the next couple hours. So on a detailed level, I'm hoping to tell you about our work with yeast prions and how this rather obscure model system has yielded some fascinating insights into basic biochemistry and human disease. And then on a more big picture level, what I hope to convey is that really almost all of the most exciting discoveries that have come out of my lab have come from questioning core assumptions that we had about the field. And usually we were wrong about those assumptions, and that is actually what yielded, I think, new directions and new insights. So before I jump into the science, I want to acknowledge two people who were instrumental in helping me develop my scientific career and my scientific philosophy. And that is my graduate and my postdoctoral mentors, Jim Maher and Reed Wickner. So these are both brilliant men, but completely different in personalities. But the one thing that I would say they had in common was that both of them were incredibly clever in having a quip for almost any situation. And so I figured I'd pick one quote from each of them that I think appropriately captured their personality. So in Jim's case, one of the things that he loved to say to us, particularly as we were preparing to start a new experiment, was that a month in the lab can save you an hour in the library. And what this was designed to point out is that it's really easy as a scientist to dive headfirst into your experiments without first stopping and going to the library and reading about what has already been discovered. And that very often meticulous planning can save you a huge amount of time of rediscovering other people's discoveries. Reed, on the other hand, I think described himself as an iconoclast and took pride in sort of poking at other people in the field. And so one of his favorite sayings when talking about some of our competitors in the field and occasionally about some politicians was that they are often wrong but never in doubt. And I love this phrase because I think it can be used in so many circumstances. I think there may be some parents, although certainly not me, who would relate to this at times. And again, listening to politicians or other people talk. But I think what it highlights, and his point with this, was that it's really important to have a little self-doubt, to question your assumptions and to not assume that everything you believe is necessarily correct. So I would say my goal as a scientist is to sort of sit at the intersection of these two philosophies, of a detailed preparation that Jim instilled and the healthy skepticism that Reed taught. Okay, so what's the field that I actually applied this to? So as we heard just a moment ago, my lab studies prion diseases. And prions, the most famous prion disease is probably mad cow disease. It was a huge issue in England in the 80s and 90s, although it's largely gone away now that we understand the cause and farmers have changed their farming practices. But there are other prion diseases. So chronic wasting disease is a huge issue here in Colorado. There's actually pockets of Rocky Mountain National Park where up to 1/3 of the elk are infected, so it's a huge number. And what these two diseases have in common is a couple things. So one is they are both infectious, invariably fatal neurodegenerative diseases. And then the second is their mechanism of transmission differs from any other infectious disease that you could think of. So every other infectious disease, whether it's a bacteria or a parasite or a virus, at its core, what allows the infectious agent to reproduce and spread is a nucleic acid genome. So usually this is a DNA-based genome. For some viruses, it's an RNA-based genome. But these infectious agents follow what we call the central dogma of molecular biology. And that is that the DNA encodes information, RNA acts as a messenger, and that messenger is then used to make proteins which allow the infectious agent to reproduce. So again, this is true for every bacteria, virus, parasite, but prions are the one infectious agent that has neither a DNA nor an RNA component. And in fact, it's an infectious agent composed entirely of protein. So this was a shocking and confusing idea when it was first proposed. It was actually proposed in the 1950s, and it wasn't until about 40 years later that Stanley Prusiner won the Nobel Prize for it. So it gives you an idea of how long it took the field to wrap their head around this concept. And the reason is we understand how DNA can be replicated. It's self-complementary, and it has a natural built-in mechanism for being copied. But that's not true for proteins. So it was unclear how a protein could replicate and spread and lead to an infectious agent. So to understand how this works, you need to first understand a little bit about how proteins normally function. So every protein in our body is made of a chain of amino acids. And so there are 20 different amino acids that compose each of the proteins in our body, and they're strung together in a linear chain. But in order for the protein to function, proteins generally need to fold into some three-dimensional structure. And so they assemble into local secondary structures, which pack into tertiary structures and quaternary structures, and you end up with this complex folded state. And this folding is driven by the properties of the underlying amino acids. So again, there's 20 amino acids, and each of these has different physical properties. So we have positively charged ones and negatively charged, and we have hydrophobic ones and polar ones. And these properties then determine how the amino acids interact. So we get local interactions between complementary amino acids that then drive local secondary structures and initiate the formation of tertiary structures. Okay, so what that means is that it's really important not just which amino acids you have, but exactly how they're laid out in the sequence, because that drives the interactions. Okay, so hold on to that thought, and now we'll talk about what goes wrong in disease. So for prion proteins, again, they have a sequence that is designed to adopt some sort of structure. But proteins occasionally misfold. And so sometimes this process fails. Usually our cells are pretty good at dealing with this. So we have pretty extensive machinery designed to degrade misfolded proteins or to help them refold. But what makes the prion proteins unique is that when they misfold, they are prone to sticking together. So shown here is a little tetramer, so 3 of these proteins that have stuck together. And what makes these really interesting is these little aggregates are actually able to bind to the normal protein and structurally convert it. So the little aggregate progressively grows and this spreads. And so it turns out these prion diseases are caused by proteins that are naturally made by our body. So we're constantly producing this protein. And if you ingest just a little bit of these aggregates, that protein will start to get corrupted and this process will spread throughout your body. And ultimately, the end result is the deposition of these very stable fibers called amyloid fibrils that are effectively irreversible. And so we have this they invariably progress and spread, and that's why these diseases are always fatal. So when I entered the field, I became interested in the question of what is it that's unique about the sequence of these proteins that allows them to do this? And at the time, there were actually 3 prion proteins that were known. There was the mammalian prion protein, and then there were 2 that had been discovered in yeast. So the same yeasts that are used to make beer and bread naturally get their own prion diseases. And they provide a really convenient model system for studying the underlying process. So I started by just looking at the sequences of the 2 proteins. And the sequences are shown here. Unless you're a lot smarter than me, I doubt you can just look at the sequence and figure out what's causing them to assemble. So this is the single letter code for the amino acids. Each amino acid has a different letter. But there were a couple things that struck me looking at this. The first was the 2 sequences are very different. So if you tried to line them up, they don't have any obvious similarity. But if you start looking at which amino acids are most common, you start to see some patterns emerge. So for example, there are 4 charged amino acids. A typical protein, these charged amino acids constitute about 25% of the sequence. But in these 2 proteins, I've highlighted the charged amino acids, they're about 5 to 10%. So they're super depleted in charged amino acids. On the other side of the equation, there are 2 polar amino acids that are very closely related to each other, glutamine and asparagine, which are Q and N. Whoops, shoot, jumped ahead. And they absolutely dominate these sequences. So in a typical protein, it's only about 10% glutamine and asparagine. These are about 40 to 50%. So this was well known in the field. So this is the Jim piece of reading what's already known in the literature. But the standard assumption was that it can't just be these compositional features because, again, protein folding isn't just dictated by which amino acids are present, but it's also the order that they're in. And the order is completely different in these two sequences. But I decided to ask, is that actually true? Is it possible that the order doesn't matter and it's just which amino acids are present? And so full disclosure, I decided I actually did this experiment as a negative control. I assumed I knew, like everybody else, what the answer would be, but I convinced myself that it was a good starting point for future experiments I planned. So the way I decided to ask this was I took the sequence of this protein and I just scrambled the order of the amino acids. So just rearranged them like shuffling a deck. And I did this, I made 10 different versions, and I was kind of shocked and appalled to find that every single scrambled version still formed prions just fine. It didn't seem to make much of a difference. And this is actually from that original paper. So this is the fibers formed by the normal protein, and then on the bottom is the fibers formed by one of the scrambled ones. And they're virtually indistinguishable. So this caused us then to completely reset how we thought about this process. And this was actually the result that I arrived at CSU with, was we knew that composition was important, but we didn't know what the rules were yet. And so I probably spent my first decade here trying to define those rules. And so what are the actual compositional features that drive this activity? And I spent a few years coming up with what I thought was a really clever way to get at this question of being able to score how good each amino acid was at forming prions. Unfortunately, when I submitted this for funding, the reviewers disagreed that it was a clever idea. And so reviewer one, I think, really nicely summed up the criticism here. So reviewer one pointed out that evolution had already done the experiment that I was proposing, and that it was not clear if these experiments would provide any novel insights. So what was meant by this was that at this point there was about half a dozen prions that were known. Again, those had developed over evolutionary time, and so if I wanted to know what makes for a good prion, I should just look at those sequences and that should tell me, right? And so the reviewer was helpful enough to go on and make a very precise prediction about what results I would have gotten if they had given me the money to do the experiments. And so what the reviewer said is that the amino acids that are common in prion proteins must be the ones that are good at forming prions. And so if we were to graph this, we'd see something like this, that on the x-axis is how often an amino acid shows up in prions, and the y-axis should be how good it is at forming prions. And so glutamine and asparagine, the fellows we mentioned before, they're right here, super common, so they must be super good at forming prions, and the charged ones would be down in the bottom corner. So fortunately, I had already started the experiments when I got this review, and I decided to continue them. And it turns out this prediction is completely wrong. So there's virtually no correlation between how often an amino acid shows up in a prion protein and how good it is at actually forming prions. Now, I must say, I was surprised by this result too. Reviewer one's argument I thought was pretty convincing. But it turns out that understanding why this is true has helped us to understand this process. So if we start looking at the amino acids, we find some patterns. So for example, I mentioned the charged amino acids earlier. So they sit down in this bottom corner. So they are very rare in yeast prion proteins, and it turns out they're terrible at forming prions. So that kind of makes sense, right? But if we look at which amino acids are common, the ones up here are really hydrophobic amino acids, ones that want to get out of water and tend to stick to each other. So it turns out that these two sets of amino acids are rare for opposite reasons. So the charged amino acids are so horrific at forming prions that if you put very many of them into a prion protein, it wouldn't form prions anymore. That makes sense, right? The hydrophobic amino acids are so ridiculously good at forming prions that you can't have very many in there, because if you did, the protein would start sticking together the moment it was made, and evolution isn't going to select for that. So it turns out there needs to be this balance where you have a bunch of these rather innocuous amino acids and a few of the hydrophobic ones that are sprinkled in that actually drive the process. Okay, so why do we care? Why does it matter that reviewer one was incorrect in this prediction? Well, it turns out that this result really helps us then understand the drivers of human disease. So to understand this, I need to back up for just a second. And discuss where the rest of the field went when we got our scrambling result. So when we realized the composition was what was so important, my lab and other labs started searching through different genomes to find proteins that had similar compositions to the yeast prion proteins, figuring maybe we could find new prions. And so this term prion-like protein was coined. And I hate the term because I think it's a little bit confusing, but prion-like simply describes how the sequence looks. It says nothing about its activity, but these are sequences that look a lot like prion proteins. And what we've discovered in the last decade or so is that mutations in many of these are linked to neurodegenerative diseases. So ALS is probably the most studied of these, or Lou Gehrig's disease. But at this point, there's about half a dozen different proteins that are prion-like in their sequence, and single mutations in these proteins are enough to cause ALS. So what's interesting is if you actually look at the mutations, so this is the chart we saw just a moment ago, many of these mutations convert these charged amino acids into hydrophobic ones. So we think these charged amino acids actually act as gatekeepers, keeping these proteins from aggregating. And if you mutate even one of them into these super sticky hydrophobic amino acids, that's enough to cause disease. And again, this didn't make sense before because it was swapping one rare amino acid for another. So what's the actual consequence of this? Well, this image shows one of these prion-like proteins in a healthy individual. So this is a muscle biopsy of a healthy individual. And the prion-like protein is labeled in red. The nucleus of the cells is labeled in blue. And what I hope you can see is that the blue and the red are largely overlaid. And so what that tells us is the protein's in the nucleus. But if we take one of these patients that has a single mutation in just one amino acid in the protein, instead we see these huge blobs of protein in the cytoplasm, so not in the nucleus anymore. And so we think that what happens is this protein's naturally shuttling in and out of the nucleus. If it has one of these mutations, it will tend to stick together. It will form these super stable aggregates that will then propagate and spread from cell to cell. And this is why we see a progressive degeneration in a disease like ALS. Okay, so the last thing that I'm going to tell you about is I think these results raise a really interesting question, and that is, why would so many proteins have these prion-like segments? So it seems a little bit dangerous to have these segments that are tipping on the edge of causing a fatal disease. And what we now think that we understand is that these proteins are actually designed to assemble. They're just designed to assemble in a weak and reversible manner. So it turns out, so just to orient you on this picture, so this is designed to show one of these proteins. PRLD stands for the prion-like domain, the part that looks like a prion protein. And what we now know is that many of these proteins under certain conditions will assemble by a process called liquid-liquid phase separation. So liquid-liquid phase separation, the simplest example of this is what happens if you take oil and water, mix them together, and shake them up. So the oil and the water will separate from each other, but they'll both still remain liquids. They're just immiscible liquids. And it turns out that many proteins are actually able to do this under certain conditions. And so this is one of those proteins that causes ALS. And at high concentrations, it will form these little liquid droplets that look very much like oil droplets. And so we think that these proteins are calibrated so that they're right on the edge of assembly, and a change in cellular environment or some other cellular cue can cause them to rapidly assemble, but in a reversible manner. So why would you want to do that? Why would you want them to rapidly assemble? Well, it turns out that this helps address a major problem that cells face, and that is that a cell has literally tens of thousands of different components, probably 20,000 different proteins and all sorts of RNAs and other stuff. And so then the question is, in that incredibly complex environment, how do you organize biochemical reactions? And one of the ways cells do this is through organelles. So when we think of organelles, we usually think of ones that are surrounded by a membrane, like the nucleus or the ER or mitochondria. And these help sequester a subset of those cellular components and help control that environment. But it turns out that cells also use membrane-less organelles, so organelles that don't have a membrane. And the nice thing about these is that they're much more dynamic. They can form when you need them. They can completely dissolve when you don't need them. Things can exchange in and out a little bit quicker. And so they're a little bit more dynamic than a membrane-bound organelle. And it turns out that many of the membraneless organelles, and this image shows some of them, their formation is driven, at least in part, by these prion-like proteins and their assembly. So we think that, again, these proteins are designed to assemble under stress conditions or change in cellular environment. And that's really nice because it allows for this dynamic reaction for cells. But the risk is you've brought these proteins close together, so in very high local concentration. And if they happen to have one of these mutations, then you've teed them up essentially to convert to the stable amyloid form, and now you are stuck with something that you can't get rid of. And so again, we think that this is what is happening in disease, that these proteins come together and they never come apart. Okay, so stepping back just a little bit more big picture, so what I hope I have convinced you of is that this formation of prion fibers itself is driven more by the composition of a sequence rather than the primary sequence. But I think what's particularly cool is this discovery that I made from a single negative control decades ago has actually driven multiple directions in my lab. So we now know that the formation, the liquid-liquid phase separation to form these membraneless organelles, it too is a composition-driven activity. So it is more dependent on the composition of a sequence than the order of the amino acids. And there are other activities that fall into this category too. So actually some transcription factors that turn on and off genes have domains that similarly are determined by the composition of the domain rather than the primary sequence. And so a large portion of my lab now is focused on trying to understand how changes in composition can determine the activities that a protein adopts. Okay. So with that, I'd like to just stop and thank the many, many people who contributed to this work. And this is only, I would say, a partial list, but I've been incredibly lucky over the years to have some fantastic graduate students. This is just a small subset of the undergrads that have come through my lab. I think these are all the ones that have published papers during their time with us. And postdocs and collaborators. We've had numerous collaborators over the years, including some that are in this audience. And funding. And then finally, I'd like to thank my department. So I think the BMB department is a fantastic and supportive place to work. In particular, I'd like to thank the BMB climbing team. So there's a subset of us that go rock climbing every Wednesday and have been an awesome source of support and experimental ideas and therapy and everything else. If you squint really hard, let's see if I can get my cursor up there, you can see me up here, and one of our other professor laureates is standing on this rock. This is Olva, and Ting Ting, I believe, is the one who took this picture. I could be wrong. But anyway, so I'd like to thank the whole department and the climbing crew in particular. And lastly, I'd like to thank my family, my biggest cheerleaders, I would say, in this process, and lucky enough to have some of them here cheering me on today. So with that, I'll take questions.
Thank you for the excellent talk. So yeah, opportunities for questions.
Is the next assumption to challenge that they are stably, like irretrievably stably deposited?
Yeah, so that's an interesting question. So for years, these aggregates have been described as irreversible, and they're not quite irreversible. So we've done a little bit of work trying to demonstrate that there is a tiny rate of exchange, but yet it's actually known there's one amyloid disease for which there's a treatment. So it's systemic amyloidosis, and it's been demonstrated with that one that if you can stop the deposits from growing, over the course of years, they will very slowly recede. So yeah, they're not quite irreversible.
I'm curious what starts the single, or what causes the single mutation that can convert something from being benign to a amyloid
Yeah, so-
platformer. Specifically in terms of ALS, where most of the disease is not genetic. We don't have, it doesn't get passed between people. It can get,
Right.
it happens like probably environmentally or something.
Yeah, so that's a fantastic question. So there's a couple pieces to it. One is you're right that only about 5 to 10% of ALS cases are inherited. And so those have been studied a lot because there we understand the initiating cause, and that can give us insights. And so the one that I talked about that we published on, that set of mutations, so that's an inherited form of ALS. But that doesn't explain the other 90%. We see similar deposits in many ALS cases. So we think that the end result is the same. Now, what causes that? There's a couple things you can imagine. So one is there could be what we call somatic mutation, meaning a single cell got a mutation, and that initiated the process. And one of the things is the mutant proteins are actually able to recruit the normal proteins into the assembly. So all it takes is one cell acquiring these mutations that could kick off the process. The problem is that's a virtually impossible theory to test because by the time you discover the disease, that first cell is long since dead. The other possibility is that we know that some of the things that drive these proteins to come together, and there is some weak evidence suggesting that those things that cause these proteins to assemble in a reversible manner, consistent exposure increases your risk of ALS. So for example, paraquat, which is a herbicide, I think, used on farms. So it causes formation of what we call stress granules that bring these proteins together, and it has been linked to an increased risk of ALS. So it's possible that every time these proteins assemble, there's a tiny, tiny risk of this happening. Mutations exacerbate that risk, or repeated exposures exacerbate that risk.
Eric, what's known about the toxicity of the aggregate sizes?
Yeah, so there's been a lot of debate about that in the field. For years, since the big amyloid aggregates were what you saw, people assumed that they were the worst piece of it. We now actually think smaller aggregates are more dangerous, I think for a couple reasons. One is they can diffuse and spread more easily. The second is these fibers only grow from their ends. So if you have a really large aggregate, it may not actually grow very well. Whereas if pieces can break off, you create new ends that can then seed the spread of the disease. So, and the other thing is the small aggregates do things like interfere with cell membranes and cause all sorts of other problems. So yeah, we now think it's actually the smaller intermediates in this process that are what are truly dangerous.
You either need to make them not nucleate or make them grow like hell.
Right, exactly. And actually, this has been a constant topic of conversation that people are concerned if you designed a drug that would break apart these aggregates, if you did it incompletely, you could actually make the situation worse. And so there's a bit of a balancing act there, potentially.
So that little experiment that you did showed that there's certain mutations that are really bad, from hydrophobic to hydrophilic, right? You would think that evolutionarily that is a driver to make sure that that mutation doesn't happen. And I'm thinking about that means that certain mappings of what amino acids you have and how that corresponds to nucleic sequence are that mapping is relevant, right? Is that mapping something that evolution chose on purpose, or did that happen just once without foresight?
Yeah, so that's a fascinating question. So just to back up to help explain for the audience, so every amino acid is encoded for by 3 nucleotides in the RNA sequence. And so that means there's 64 possible combinations, and they determine which amino acid you make. And so the code that translates RNA into protein is actually set up so that most mutations will make rather innocuous changes. So we'll change one charged amino acid into another charged one. And so if you actually look at how the amino acids cluster in the chart, most mutations are reasonably conservative. but there are exceptions. And so, so these tend to hit the exceptions that aren't changing one negatively charged amino acid to another negatively charged amino acid, but instead are dramatically changing the properties. So yeah, I think my guess is evolution has selected for reducing the probability that this happens, but you can't completely eliminate it.
So you mentioned at the beginning that mad cow disease has kind of died out. Do you know what causes such a disease to die out?
Yeah, so mad cow, most mammalian prion diseases really are not very infectious. So they are infectious if you try really hard. For example, the first human prion diseases were identified in a cannibalistic tribe in Papua New Guinea. So they had a ritual of eating their dead, and that's what caused the disease to spread. But they do not spread by proximity, most of them. Mad cow disease is the same way. So if you could put an infected cow and a healthy one together forever, and it's not going to spread between them. So what spread the prion diseases in England was that as a way of beefing up cattle stock, they were feeding rendered cattle parts to other cattle. It turns out this is a really good way to spread a disease among your cattle. And so presumably one of them got infected, probably crossed over from sheep, we think, and then essentially fed to his peers, and it spread very, very quickly. And it takes a while. The incubation time is years, so it had spread pretty widely before it was even identified. So you just stop that farming practice and it goes away. And that's what happened. It's amazing just how it plummeted over time. So it'll still spontaneously pop up every once in a while. Every year or two you hear about a new case just sporadically, but it doesn't spread. Chronic wasting disease is the one exception. It is super infectious, and we don't know why. We think it's a fecal-oral route that it spreads from one deer to the next. So, a deer defecates on the grass, another deer comes along even a decade later and eats from that spot, and they'll get sick. And so it's incredibly stable, persists in the environment, and can spread. Yeah.
A couple more, and then we're going to have to.
So from my knowledge about yeast prions, they aren't like disease related. They have beneficial purposes of memory for mating. Do we know of any similar positive role that prions might have in humans, or are they all infectious, disease related?
Yeah, so that's an interesting question. It's actually a huge source of debate. So the conventional wisdom in the field is that actually yeast prions are a beneficial regulatory element, that it's a way of switching the activity of proteins on and off. Reed, my postdoctoral advisor, would say that that's one of the areas where people are often wrong but never in doubt, that he is adamant that that is not correct, and that they're diseases of yeast. So we don't know for sure. We do know that there are prion-like proteins that form amyloid aggregates that are actually beneficial in humans and other organisms. So for example, it's thought that there's a prion-like conversion that is involved in memory storage. So we get these super stable aggregates, and that actually is a way to help memories persist. But there's now been a number of these beneficial regulatory elements that have been identified. Yeah, good question.
So one of the things that I'm impressed by in this talk is that, and it's becoming more and more apparent, and I have a background in protein crystallography, is that partially or maybe even loosely folded proteins perhaps are the norm, rather than the detailed 3D structures that we have been learning about and spending the last 50, 60 years working on. I wonder if you had any thoughts about that, about whether, we really don't know much about biochemistry because we've been so focused on the 3D structure of proteins and their role in the various things that we do attribute to 3D structures.
Yeah, so that's a fascinating point. It's actually one that I sort of cheated and glossed over, I would say. So what the question alludes to is that about, I think, 30% of the human proteome doesn't actually have a defined stable fold. It's called intrinsically disordered, meaning it can adopt different structures. And actually, the portions of these proteins that drive prion activity, they are intrinsically disordered themselves. So they don't tend to form a single stable fold, and that perhaps frees them up to accidentally stick together when they're not supposed to. And big picture, I think, with your question, yeah, I think it's historically in the field, we focused on the things we can see. And with crystallography and other methods, you can see the structured domains. And so we've tend to ignore the disordered stuff. But it's there for a reason, and it likely drives very important functions. And so yes, I think that there's definitely been a migration in the field of recognizing that these disordered segments can drive really critical activities, including this liquid-liquid phase separation to drive protein assemblies.
All right, well, on the theme of cutting off interesting conversations, let's go ahead and thank Dr. Ross again for an excellent talk. Sneak up and say one more set of things. Okay, I'd like to congratulate all our award winners once again tonight. And I'd now like you to invite all of you to join us for refreshments and to visit with the Professor Laureate and the awardees. So please join us for reception.